Hello, I'm Agostinho Walter, and together with Pablo Campuzano, Diego Hernandez, and our mentors Camilo Ayala Garcia and Giovanna Danies, we are exploring the world of microbial fuel cells. The microbial fuel cells we are building capture electrons from anaerobic exoelectrogenic bacteria, which live in the soil, such as Geobacter or Schwannella oneidensis. The research we reviewed in microbial fuel cells focused on efforts to optimize the electrical energy harvested from the bacteria. For a time, we also tried to investigate harnessing this power, but in working with the bacteria, we realized that the electricity generated on their part has more use as a signal for the communication than as a source of clean energy. The electrical energy being produced changes based on the composition of the local ecosystem that the bacteria are part of as decomposers within the soil. In man-made ecosystems such as compost, this electrical energy could translate to hidden insights about our society. Here in Bogota, Colombia, there is an initiative in our communities to reduce the amount of waste going to the Doña Juana, the principal landfill of the city. This initiative uses the Pacadias Torre method to dispose of any and all household organic waste. Banana peels, eggshells, chicken bones, you name it. What happens is at least once a week, usually Sundays, community members meet at the Paca to compose together the waste that's accumulated over the week. Since the process is labor-intensive, the more the merrier. The Pacadias Torre is simple in principle, a homegrown Colombian invention. The compost pile begins with a layer of branches, and successive layers of organic waste in nests of foliage fill the paca. Compressing the layers as much as possible is of the utmost importance. This is what creates the hermetically sealed interior that allows anaerobic bacteria to thrive. The same family of bacteria use the microbial fuel cells. Anaerobic bacteria differ from the aerobic bacteria used in traditional composting. Unlike aerobic bacteria, anaerobic bacteria can break down all organic matter without odors, following the natural cycles of decomposition beneath the forest floor. After 6 to 12 months, all of the organic waste turns to humus and fertile soil, teeming with plant and animal life. This virgin soil can be harvested and used to cultivate agriculture, such as the lulo and the urban environment. These pacadillas torres have appeared in each neighborhood of Bogota and in multiple cities of Colombia, including Medellin and Cali. For the founder, Guillermo Silva, the real value of the project is to build community. Decontaminating the environment is of secondary importance. In the Paca we are a part of, we have seen this to be the case. On Sunday mornings, the Paca is a place where the community of the neighborhood of the La Macarena comes together to offer our organic residues to our bacterial and insect neighbors, while trading news and knowledge over enjoying arepas and chocolate. We believe that Paca offers more than just a way to dispose of our waste using our natural ecosystems and fortifying social links. The Paca offers a unique ecosystem that is based on a man-made energy source, our waste, making the resulting ecology a reflection of our alimentary practices. In communities that consume meat, the Paca will see more bones, while others that have more fruit and vegetable-based diets will create Pacas with a more plant-based composition. The difference in Paca composition leads to different insect life within and a different microbiome. We theorize that measuring the electrical output of exoelectrogenic bacteria in the microbiome of the Paca could be correlated to the diet of the community, and thus be a marker for community health. To investigate this claim, we are conducting a three-pronged investigation. We are doing a comparative study between the electrical output of different soils produced by Pacas at different periods, May 2020, August 2020, and October 2020, to see how the production of electricity varies among these different soils from different time periods. Additional work that could provide useful data is to measure other markers of soil, such as the insects that live within and the bacteria that make up the microbiome, and to analyze the nutritional content of the soil as well as the pH. B. We are observing how the electrical output of soil changes over time as a function of humidity, temperature, and the ecological systems such as the type of plants and insect life present. C. Using these insights, we are designing and prototyping a material sensitive to the electricity produced by the bacteria, which can translate the electrical signals for the bacterial community. Depending on how the actuators are attached, the material can translate the measured voltages to a visual output to show the health of the Paca as the organic waste decomposes through color changes of LEDs or the numbers of LEDs changing. This instrument can inform the community of what's going on underneath the surface of the paca through seeing the metabolic activity of their bacterial friends and running it back to what they ate and threw out, and draw conclusions about how their eating habits create different ecologies and what these ecologies mean about their community health. This can only last up until the paca sinks into the ground and is harvested. After this point, in private, members of the paqueros can place soil from the paca into the material and see how the electrical energy is translated into kinetic motion and can interact with the bacteria in the soil, gaining an embodied knowledge of their community health based on the ecosystem and the soil from this period. By placing soils from different time periods, paqueros can interact with the bacteria from different time periods of their community, reliving Christmas memories with the bacteria which arose from the ecosystem created by the Christmas ham of yesteryear. This relationship which we build between us and the bacteria could be another one that integrates us with our environment. Just as we have meaningful relationships with our pet dogs and cats, or the wild hummingbirds that frequent our gardens, we can build a relationship with the microorganisms that owe their existence to what we deem waste, and expand our consciousness of our world further. The material that we propose, which can detect these bacterial signals, can translate these signals however the user wishes in order to build a dialogue with the ecosystem. While we built a version of the material using decomposable cotton cloth, a pillowcase, and electrodes of stainless steel mesh coated with activated carbon and carbon black. In reality, the platform for the dialogue can be of anything, as long as it contains the electrodes which are sensitive to the signals. Previous iterations include thermoform textile, which could also be decomposable. Considering the low cost of these materials and the open source nature of the project, we encourage that this project goes beyond the paquero communities of Colombia and evolves to fit other contexts that go beyond understanding community health and its history on a personal scale through microbial archaeology of the ecosystems that spring from our food waste. The potential of exoelectrogenic bacteria is more than their voltage. Their production of electricity allows them to interface with electronics and computation, creating a bridge between natural ecosystems and the digital. We are excited to see where that bridge can take us.